Okay. So let's have a class. And the first lecture, which is lecture 1, exam 1, is always about the history. Now the first question, what is microbiology? There's a lot of definition talk about microbiology, but I will say, very simple, this is science behind microorganism. So what are the science we're going to talk about behind the microorganisms based on the syllabi we just talked? Basically is isolation, identification, and numeration of microorganisms, including microscope use, staining technology, and some biochemistry identification. The second thing what we're going to talk about is disease caused by different microorganisms. And here we're going to focus on is a bacteria caused disease. Will be the major part of this semester's material. We will have a lot of case study about that. I really like it. It's coming from the real life, from the CDC website. Then to talk about these pathogen-related case, and you're going to identify which pathogen causes those diseases. Number 3, this section, lots of you have been learning before, already learning, or is being learning right now, which is about biochemistry and genetics. So biochemistry, we will talk about ATP, which means energy generation. That's energy calculation will be major part of this section. We're going to talk about glycolysis, TCA cycle, and fermentation. Genetics part, we basically talk about DNA, replication, transcription, and translation. And a little bit of regulation, we will folks talk about lacto-alpha. So this is the third part. Now, lots of you, when you go to that stage, they are feeling a little bit bored and say, I already learned in the other classes. Why are you going to talk about here? Because here we're using bacteria, usually E. coli as an example. And when you learn in your biochemistry class or the biology class is using eukaryotes, animal and human being as an example. So there is a slightly difference, and we will mention. So what you learn in your biology class is certainly helps, but here will be a little bit different, and you still have to listen in carefully. Okay? Now last section, applied microbiology. So I'm a food microbiologist. I do lots of things for the pathogen control. We certainly will talk about foodborne illness. In the West Virginia area, the poultry production is very popular. Although we are not a poultry state, so we will talk about the mobile poultry processing unit, MPU. It's typically in West Virginia area. And we will talk about immunology, because immunology is part of the microbiology section. And the last thing I always put at the last, we will talk about one lecture for virus, because you heard about too much during the COVID-19. You're pretty much like tired of that. So we put at the end, because I just want to give you refresh information regarding the virus, what it looks like. So this is microbiology, the science behind that. Okay, so what is microbiology? The next thing what we want to talk is, what is microorganism? So this word is microorganism. I think it's a very nice word because we usually can split them right in the middle. MICRO is micro. What is micro? It's very small. And based on the slides, you can read, it said, too small to see. By naked eyes, so by eyes. So what we have to do, we need to use a tool, the tool called microscope. Therefore, in this classroom, especially in the lab section, we will practice microscope use a lot. Now, there is a lot of microscope. There is a light microscope. Light microscope also simply become bright field and dark field. And then we have like laser confocal microscope. What's the difference between that? Light microscope, the energy resource is light, and the laser confocal microscope, the energy resource is laser. We will talk about that later. What else we have? Fluorescence microscope. Fluorescence microscope. Fluorescence microscope, we usually is put a dye, a fluorescence dye. It will show you a shiny fluorescence color, and you can identify the bacteria without doing any other thing. And you can differentiate live and dead cell. That's very important. Live versus dead cell. Okay, now what else we have? Light field microscope, laser confocal microscope, fluorescence microscope, of the last one, electronic microscope. Electronic microscope will introduce transmission electron microscope and scanning electron microscope. One to see the internal structure of the microorganism, another one is to see the 3D surfaces. And we will introduce that. But in the lab section, because of the conditions and the time limited, we do not have these things to do. We will focus on light microscope and focus on bright field microscope. Okay, so this is a microscope. I just gave you some introduction. So we talk about the micro. It's very small. It's too small to see by the naked eyes. So what does the organism mean? Organism means live body. So what is microorganism? Is a too small to see live body. And we'll talk about these one by one, give you some examples. So the first question, how many microorganisms in the world? Can you guess? Thousands, millions, trillions? Some people make a joke using gazillions. It's a word from kindergarten. We talk about this too many. How many? There is no definite answer for that, but there is a scientific survey saying in the world there is about 10 to the 30th microorganisms. The number is huge. This is the first thing. Second question, how many many microorganisms we already know? Are we know all of those? Obviously not. We know about 5%. So there is about 95% microorganisms are unknown. This is the reason we have COVID-19, these bad bugs comes out. And nobody is prepared. Remember 3 years ago, you just came to the WVU, you stayed in the dorm for the whole semester for the online classes. It's pretty boring. We never prepared because that bacteria, that's a virus, we don't know about it. So 95% are unknown. And I also want to tell you, there is some scientific research saying, so predicted that maybe like every 15, 20 years we have something comes out same like COVID-19, the people have to be prepared. Because a huge number we don't know about that. And we know a very limited is 5%. Okay? Okay, now, what are the examples of microorganisms? We are talking about so many. Well, let's talk about it. First thing first, the microorganism. You know bacteria. You learned about when you were very young. Bacteria, bad bugs. What are the examples for bacteria? Did you hear the E. coli? Yes, of course you heard about it. And I often make a joke. I said, even if you failed AEM 341, although it's never happened, and even you get, like, say, C in the classroom, and 30 years later, you still remember one bacteria name is E. coli. But there's a lots of E. coli, so we will talk about it. Did you heard about salmonella? You heard about salmonella. It's a pathogen in the poultry meats. Did you eat those breaded chicken nuggets when you were young? You like it in chicken fillet? Those breaded chicken nuggets right now is 0 tolerance for salmonella. So which means we do not allow one cell of salmonella to be tested on the breaded chicken nuggets. It's a new regulation by USDA FSIS, United States Department of Agriculture and Food Safety Inspection Service. So that's salmonella. Did you heard about listeria? Yeah, you heard about that. Back in 2012, there is a big cantaloupe outbreak about Listeria monocytogenes. And very special for this bacteria can grow in the refrigerated temperature. Usually, lots of the bacteria cannot grow at lower refrigerated temperature. So that's the first thing, is bacteria. Okay, second thing, fungi. You heard about fungi. We have major 2 different categories, yeast and molds. And we will mention that later on in the one section of the lab. Yeast, 37 degrees Celsius grow, molds 25 degrees Celsius most of the time. Do you know about molds? You had a bread in the breakfast, and you left in the dorm on the bench. You didn't finish it, you rushed for the classroom studies, and you didn't clean it up. 7 days later, in the room temperature, you see those greenish color there. That's molds. Really bad, don't eat it. There is a toxin there, alpha toxin, could be. Okay, that's molds. Yeast. Do you like beer? Everybody likes beer. There is, some people don't like, that's fine. And it's an alcoholic fermented product. What's a pathogen? What's the yeast for that? Saccharomyces cerevisiae. And in our first day lab, next week, we will do the observation under microscope. So that's yeast. Okay. What are the other examples? Protozoa. It's a separate class at WVU, is that right? Animal nutrition science. We have a separate class talk about protozoa. What are the examples? Did you heard about Toxoplasma gondii? Toxoplasmogonia has a middle host, which is a cat. And so the pregnant woman will not be recommended to have a cat at home because it could transmit it and then cause miscarriage. That's Toxoplasmogonia. It's an example for the prototype. Okay, what are the other examples? Algae. Did you heard about that? This is I usually call it an environmental microorganism because it's responsible for 75% of oxygen in the world. So that's environmental microorganism. And very important, this microorganisms, they use sunlight as an energy resource. Let's say E. coli, their energy resource is sugar, is glucose. If there is no glucose, they have to use lactose and sucrose and other alternative sugar. But for the algae, what they do is sunlight, because they are doing photosynthesis. So photosynthetic bacteria, it's a little bit different. Okay, we will touch base a little bit, but not too much. What else we have? We call it slime modes. And this guy is very tricky. Slime modes, they have two growth phases during their lifetime. Part of that, like a protozoan, and part of that, like a modes. And what they do, this is an environmental cleaner. So let's say you have a leafy there. This is a debris, and dust, let's say. And this microorganism, they could be engulfed with. And then lysis, and they eat it. But I will not say where they eat. I will use quotation mark and eat. So that's called the slime modes. Now all of these in your slides, which is gave you a name, which is called protists. So that's all these three different categories. The last one, archaea. A-R-C-H-E-A-E is archaea. I will say this, archaea, it is underdeveloped bacteria. So for example, you learn in your other classes, bacteria has a cell wall. Has a major components of the cell wall is peptidoglycan. And archaea, they have it, but it's a different name. Another example will be bacteria moving needs to have a flagella. So let's say this is the bacteria, the flagella is right there. The bacteria is moving, it's a real moving, okay, called the flagella, the structure. And the archaea, they do not have a real flagella. They're a little bit different. So we call it an endoflagella. So lots of the structure very similar to the bacteria, but we gave them a different name, and we call it underdeveloped. A good example in your slides, which is methane, because this environmental archaea will generate methane gas, CH3. The methane is important. Remember where they come from? In the fecal samples. In the older time, there's a big pond, they put methane there, and do the fermentation will generate energy. And that energy could become electricity. That's a very old time to do that. It's called energy transfer. And we will mention something in our exam sheet. Okay, so all these, we put them together. We call them cellular, because they use cell as a single unit. In other words, there is a different category which is not a cell as a single unit. We call it acellular. So what are the examples? Number one, virus. So we want to talk about a little bit right now, and we'll talk about a little bit later on. What's the structure of the virus? And we will talk about structure of bacteria in the lab and the lecture section later on. It's very complicated. The cell wall, the peptidoglycans, the outer membrane, you have to draw it. Do we have some question for you? It's pretty difficult. The structure for virus is very simple. What it looks like. Basically like that. What are the inside? Nucleic acids. It is DNA or RNA, but never both. So what are the outside? This structure we call it a capsid. It's a protein. Therefore, it makes antivirus is very difficult. It's a challenge. Why? If you have some drugs, is anti-DNA, RNA replication, transcription, translation, they will generate lots of mutations. So you always remember, if something attacking a microorganism is for DNA and RNA, sometimes doesn't work very well because lots of the mutations will generate. But let's say it's attacking protein. Is that good? Not really, because human beings, animals, even plants, they have proteins. So when you're attacking protein, you are attacking the same person. There's a very strong side effects. Okay? So when we talk about the coronavirus, what it looks like? If you have these, like a crown, looks like that, so we call it COVID-19, so coronavirus ID 2019, because it's found December 2019, although the outbreaks happened in 2020. When you see these spikes there, is that right? It's a corona shape. They're going to shift it. So what's going to happen if they shift it? They will generate lots of mutations. So what are the mutations you heard about? Delta, you heard about it? Which is really the bad one is Omicron. Is that right? Because millions, thousands of cases every year. But right now, nobody talk about that anymore. Well, part of the reason is the newsletter and the news media cycle. But another thing I also want to mention, we had a very good system for the vaccination. We have to say that. And the vaccination is the best way to prevent virus disease. And most of you have a shots, is that right? I have from Madura, have a first and a second shots. Then some of you recommended for boosters, and I have a third and a fourth boosters. So Pfizer and Madura, you have to do different booster shots, and the Johnson & Johnson is only one shot. Well, different people have a different opinion about the vaccination. We'll talk about that later on when we move to the vaccination section. But scientifically, you have to recognize the vaccination is a way, I would say the best way to prevent the virus disease. Because the drugs usually have a very strong side effects. What are the other ways we can prevent a virus? We call it antibody. So do you remember, before the vaccination of the coronavirus comes out, what they do? Some hospital in China, part of the hospital in the United States, they get the patient blood sample. Then they do the centrifuge. Then they get the saline, is that right? The saline, which is a liquid part of a blood without red, white blood cell. It's very light liquid. They have a lot of antibody. And then sometimes you can inject it to the patients, and you can do part of the treatments. So that's another way to do is antibody. But I want to tell you one thing. Isolation and antibody is very difficult to do. We will mention this later on. It takes lots of time, and the work is tedious. So usually, not many products about that to use antibody to do the treatments. Although scientifically it works, but practically sometimes doesn't work. And most of the time, we actually using antibody to do the testing, to find the bacteria, to find the virus. So we will talk about that later. So this first thing, virus. What else? Varroils. Varroils is a single AEM. This is caused a very severe plant disease. Back in Philippines in 1996, there is a flood area. All the coconut trees has been damaged because of the varroa. So that's varroa. Number 3 is called a satellite. I will put a quotation mark there. I never heard about that. We're not going to see in the lab. It's just in the slides, in some of the textbooks. This is some people say it's an RNA engaged with protein. A protein is like a coat. So we say it's engaged with a protein coating system. That's called a satellite. That's all. We're not going to talk too much about that, because I never heard about that, we don't have an example. Last one. So we mentioned about a virus. You have a structure of DNA or RNA, which is genetic information, and they have a protein. So we mentioned varroils, which is single RNA, and the satellite, which is RNA engaged with protein. But how about single protein? Is that protein could be a trouble for us? Yes, they're called a prion. That's a protein. It will cause neurological disease. What are the examples? Did you ever heard of medical disease? This happened in 1997 in Great Britain, in the cows and even cattle. It's a brain system damaged, including the immune system, can transfer to the human being. It's a severe damage, and you can only do is deep bury. Because of that, even today, Australia, Russia, and China, they still forbid the United States beef to import it there, and even a beef jerky, all those beef products, because of the major concern of the medical disease. And that's caused a lot of damage for the cattle and the beef industry in the United States. In the human being, there's one disease called Kulu. This is a very interesting. What is Kulu? It's a neurological system disorder caused muscle relaxing. So what's going to cause a muscle relaxing as a result?